to the highly advanced nature of many modern cancer treatments, the pharmaceutical industry is faced with a drug discovery challenge in how to successfully select the most reliable and translational in vivo models for their preclinical testing of new oncology therapies. So many drugs fail in late-stage development, and here oncology actually holding one of the highest preclinical to clinical drug failure, with 95% of preclinical drugs failing during clinical development. So how can our platform assist in these challenges? Well, we have developed a zebrafish tumor xenograft platform that we called ZTX. And our platform can predict treatment response in patients with very high accuracy. So our model is providing a highly translational model for preclinical testing of new oncology therapies. Our model provides functional in vivo data on the efficacy of anti-cancer compounds on reduction of primary tumors as well as metastasis in only 5 days. The model can be used throughout the whole preclinical to clinical pipeline, as you can see here in the slide that I'm showing. So during the preclinical phase, the model can be used for target validation and lead optimization and screening both compounds and tumor indications, selecting the best leads from compound libraries or different formulations to take further into mouse PDX models. Using the ZTX test during the clinical stages, it can be used for patient stratification for clinical studies to find which patient subpopulation that show the best response to the specific compound. The test can also be used as a companion diagnostic for specific compounds. And finally, in the clinic, the test is used for selecting which treatment that should be given to specific patients. Here we screen the different treatment options, providing results to the treating oncologist or surgeon, which treatment that is most efficient for this specific patient. When it comes to the clinical aspect, we get tumor samples directly from the surgeons in the operating room. These tumor pieces are sent to us here at Bioruperia. We then treat these with an enzyme mix, giving us a single cell suspension consisting of all the different cells that this patient tumor consists of. We then label the cells with this red fluorescent dye and inject into the zebrafish embryo. We then treat the embryo with the same treatment that the patient could be given throughout a period of 3 days. And we then evaluate how efficient the different treatments are, and being then able to send a report to the treating oncologist with the scoring on how good these different treatments are, aiding them in their selection of which treatment that this specific patient should be given. I will now go over to show you some clinical data that we have obtained from our platform. So I will start with our first finalized pilot study, where we have evaluated effect on patients with muscle-invasive urinary bladder cancer. So the standard treatment plan for these patients is that they undergo a surgery called TURB, where most of the tumor is taken out. They then undergo 4 months of neoadjuvant treatment. And this is a cisplatin combination treatment. After these 4 months, these patients undergo cystectomy, meaning that you remove the whole urinary bladder. This treatment is highly efficient for 35% of the patients subjected to it. And this is mostly due to that they are responders to this neoadjuvant treatment. However, 65% of the patients do not respond to the neoadjuvant treatment. This means that after this whole period of TURB, 4 months of neoadjuvant treatment, and the whole recovery process after the cystectomy, the treatment needs to be stopped and a new decision on which treatment to apply will be taken. If we use our platform, we can already, after this TURB, evaluate the tumors inside the zebrafish embryo, subjecting them to the same treatment that the patient will be given. We will then in 5 days find which patients are responders and non-responders to this treatment. The responders will go into the exact same treatment plan as the one above, with a surgery, with a standard treatment plan, and will have a very good prognosis. But the non-responders will then also be evaluated according to their metastatic prognosis and be decided, will they have immediate surgery, or if they are prone to develop metastasis, a very fast decision on a new treatment. Remember, this is only 5 days after this initial surgery. And with this model, we will have a good prognosis regardless of which response you have to this initial treatment. So we have conducted a pilot study where we have compared data in our platform to the actual clinical data from these patients. And 50 patients have been included in this pilot study. And we can see that for treatment outcome, our platform shows treatment correlation of 80%. So we can find 80% correct when predicting responders and non-responders to this cisplatin combination treatment. Also, for predicting metastasis, we can show a clinical correlation of 100%, meaning that we can find all the patients that will or have developed metastasis during this neoadjuvant treatment period.
For more information, please visit our website or contact us directly to set up a meeting to discuss how the zebrafish can help you.